Okay, so we're going to start just with a brief overview of the endocrine system, which is the system that controls hormones in your body. So, what is the endocrine system? So, the control system of the body that is slow acting and long lasting when compared with the nervous system. So, the nervous system, if you remember from the past lectures, is a very quick acting system. It uses electrical impulses. It can transmit information throughout your body, from one end to the other in a fraction of a second. The endocrine system, in contrast, is slower acting and it's more long lasting. So it has a longer lasting effect on the target tissue that it reaches. So, like I said, while the nervous system signaling is usually mediated by electrical impulses carried by neurons, endocrine signaling is carried out by hormones released into the blood and circulated to other tissues. So, that released into the blood is a very important part of the definition of what an endocrine molecule is. Hormones are released into blood. There's another term that we'll talk about in a little bit called exocrine, and that means that it's released into a duct that's not actually released into the blood. But anytime you hear endocrine or hormone, by definition, they have to be released into the blood to circulate to other tissues. So endocrine signaling can be preceded by nervous signaling, and it usually is. And we're going to talk about the HPA axis later. That's basically how your body reacts to stress and how it bridges your nervous system and your endocrine system to create a holistic response. So the endocrine system uses a variety of types of signaling and a variety of hormones to affect the body, obviously. So there are a bunch of different hormones and there are a bunch of different receptors on all of the target tissues. But there are actually different types of signaling that the endocrine system can use. So I put those on this slide over here. So we're just going to go over them. So there are 4 types of endocrine signaling that you should be able to identify for the MCAT. So you definitely want to be familiar with all of these, at least the first 3. So the first one is just straight endocrine signaling. So the target tissue is not in close proximity to the endocrine tissue. An example, insulin is produced in the islets of Langerhans and the pancreas, and it circulates throughout the body to interact with distant muscle tissues. So that's what straight endocrine signaling is. That's basically just the definition of what the endocrine system does. The target tissue is not close to the specific cell. It releases hormones, enters the bloodstream, and it can circulate to a farther distance and interact and affect change in that tissue. The second type is paracrine signaling. So the target tissue is in very close proximity to the endocrine tissue. So it's endocrine signaling, but it's just closer. These paracrine factors are released directly into the extracellular matrix instead of the bloodstream. So an example of paracrine signaling would be the release of neurotransmitters into the synaptic cleft. So they're not touching each other and they're not communicating with each other via electrical impulses. They're communicating with each other through this release of signals. So, and because they're so close to each other, it's considered paracrine signaling because that's how one molecule is relaying information to the next, one cell, excuse me. The third type is autocrine signaling. So the endocrine tissue also expresses the receptor for its hormone. So the cell that creates the actual hormone is also expressing the receptor and is also receiving the hormone. So you could kind of see how that becomes sort of positive feedback loop, and it could be, it's okay if it's in a controlled environment, and it could also be problematic if it's not controlled. So this type of signaling is often found during growth and development, like embryonic development. I think early childhood development, there's autocrine signaling in certain types of cells. The specific types of cells are not too important. It's just, you should know that autocrine signaling is really mostly found in development and in your immune system. So during T cell activation, the T cells will actually release factors that it actually expresses a receptor for. So it results in what we call monoclonal proliferation, which is, it just makes a bunch of clones of itself, like I said, in that sort of positive feedback loop. So this type of signaling is also often found as a pathway for cancer growth. If you've ever taken a cancer bio class or you've ever studied cancer, you'll see that a lot of cells, one of the ways in which they evade these natural checkpoints and the ways which they continue to grow despite what the body thinks is natural is that they take advantage of this autocrine signaling pathway. And that could lead to tumor growth and eventually metastasis of the cancer. So the fourth type of signaling is not one that I've seen very often in my own studies, but I did see it a couple of times upon revisiting when I was making the lecture. So I thought it was at least a good idea to include it. And there's not a lot of these types of signaling, so I think it's probably a good idea that you guys do know it for the MCAT. So it's called Juxtaprint Signaling. It's also known as contact-dependent signaling. So it depends on the type of connection between two adjacent cells. So it seems kind of similar to Paracrine signaling, but Juxtaprint signaling, the cells are actually touching each other. So there are three different types. Juxtaprint signaling can be broken down into three types of signaling, and I put it right under that. So the first one is cell-to-cell signaling, and the ligand of one cell's receptor is found on the membrane of an adjacent cell. So one cell, the membrane is expressing a protein that's maybe sticking out of the membrane, and that protein actually hits the receptor of another cell, and that's considered juxtaprint signaling. Gap junction signaling, two adjacent cells communicate by a direct connection. So think back to electrical synapses when we were talking about the nervous system. That's a form of gap junction signaling because these two cells are connected between these gap junctions, and they can communicate with each other that way. And then the third one is cell extracellular matrix signaling. A cell's receptor is activated by glycoproteins found on the extracellular matrix. So it's similar cell to cell, but it's just cell to extracellular matrix. So there's some sort of protein or glycoprotein that's in the extracellular matrix that is interacting with the receptor on a specific cell, interacting with a specific receptor on the cell membrane. And this is one of the ways that cells like interact with each other. And just spread communication with each other. And it's important for like cell proliferation and making sure that none of that goes out of check. Okay, so now we're going to talk a little bit about the different types of hormones. Hormones are generally derived from three different types of precursor molecules. So the first one I want to talk about are lipid-derived hormones, and in humans, it's primarily the steroids. Most of these hormones are derived from cholesterol and are lipid-soluble, which means they're hydrophobic. Sometimes they'll say hydrophobic, sometimes they'll say lipophilic. It all means the same thing. They're just nonpolar molecules. And if you look at most of these molecules, they have very similar structures to cholesterol. So if you know what cholesterol looks like and they give you the structure of a hormone and they're asking you to identify it, it might be pretty useful to know the structure of cholesterol so you can easily identify them. And lipid-derived hormones can diffuse through the cell's phospholipid membrane to act on interior receptors. So remember, they are hydrophobic, and so is the phospholipid membrane. So these cells are able to diffuse straight through without any sort of transport protein or anything like that, and they act inside. So since they're not water soluble, like we just said, steroids rely on transport proteins to get through the blood. So albumin is one of the bigger transport proteins that you find in the body. And they usually will just like via nonpolar interactions, they'll just stick to albumin and they'll circulate the body. And then once they get to the target tissue, they will dissociate from albumin, go through the membrane, and they'll enter the cytoplasm. So steroids often affect the body slowly since they directly affect RNA transcription. So a lot of steroids act directly on RNA transcription, and that's their function. It's not like a peptide hormone, like we'll talk a little bit how it alters the enzymatic, like the enzymatic stuff that's going on inside the cell. These actually directly affect RNA transcription and what is being transcribed. So because of that, their effects can persist for weeks in the body. And they're synthesized in the Golgi on demand, and they're shipped immediately since they can't be stored in membrane-bound vesicles. So the vesicles in your cell, they're very similar to the cell membrane. They're hydrophobic. They're usually made of phospholipids. So we just said that steroids can't, they're hydrophobic, so they go through these membranes. So we can't synthesize steroids and store them because they'll just leak out. So that's why we actually make them on demand. They're synthesized in the Golgi and they're immediately shipped out. They're very short lasting and they affect their target cells very quickly. And a little bit about the nomenclature. Steroids will often end with the ONE or OL suffix. So we have estradiol, which is an estrogen, testosterone, aldosterone, and cortisol. Those are all lipid-derived hormones. They're all steroids that that we find in the human body. And we'll talk a little bit about specific hormones later in the lecture. Okay, so the second type of hormone that I want to talk about are peptide hormones, which are probably the ones that we're most familiar with because we talk about a lot of them. So these hormones are derived from peptide precursors, and they are produced in the rough ER. Obviously, because the rough ER is where where most of the cell's ribosomes are. So the peptide hormones are mostly produced in the rough ER. So peptide hormones, unlike steroids, are hydrophilic. They are polar and they can dissolve in water. Therefore, they cannot diffuse through membranes like steroids can. So also, since they can't diffuse through membranes, our cells are able to store them in vesicles. So these types of hormones are often produced in advance, and they're stored until they need to be utilized. So make sure you understand that conceptually, that peptide hormones, we can create them in advance, then we can store them. So we can release them pretty quickly once we need them. The steroid hormones we have to produce on demand. So it takes a little bit longer because we have to produce them, but we immediately ship them out because we can't store them. So peptide hormones, since they are hydrophilic, they can dissolve in the blood plasma. They don't need transport proteins because they are proteins. And they are transported to receptors on the surface of the cells, on the surface of the target cell's membrane. So they don't diffuse through. The receptors are on the cell's membrane on the outside, sticking out. So peptide hormones bind to a receptor. The hormone initiates a signal cascade inside the cell, and that alters the existing enzyme activity in the cytoplasm. The effect of the peptide hormones are relatively quick, and they begin minutes to hours after secretion. And peptide hormones often, but not always, end in IN. So that's a pretty common protein ending. So like insulin, for example, is a peptide hormone. Oxytocin is a peptide hormone. But then again, follicle stimulating hormone is also a peptide hormone, and it doesn't end in IN. So if you see IN on the exam and they're talking about a hormone, it's safe to say that it is a peptide hormone. But the other ones, just because it doesn't have an IN doesn't necessarily mean it's not a peptide hormone. And FSH is a pretty common one that you'll see on the MCAT. So you should commit to memory the fact that it is a peptide hormone. And the third type that we're going to talk about are amino acid derived hormones. And those are the hormones that are derived from single amino acids, whereas peptide derivatives are created from polypeptide precursors. So there are multiple amino acids that are hooked up, and those are the ones that create peptide derivatives, but the amino acid hormones are just, they use just singular amino acid precursors. And since the hormones are so small, they can take on properties of the other two groups of hormones, depending on where they're synthesized. So some amino acid hormones will act like peptides, and some of them will act like steroids. So the catecholamines, epinephrine, for example, these are the hormones that are secreted by your adrenal glands. The catecholamines don't go through your cell membrane. They actually bind to cell surface receptors, and they're pretty quick acting. So in that way, they're very similar to peptide hormones. T3 and T4 are two of your hormones that are secreted by the thyroid. They have an iodine component, and they're actually able to diffuse into the cell, and they act on interior receptors, a lot like steroids and lipid-derived hormones that we were just talking about. So both of these catecholamines and the thyroid hormones, they're both amino acid-derived hormones, but you can see they act very differently. Their behaviors actually resemble those of the other two categories. But for our categorization purposes, we divide them into three based on their precursors. So that's a pretty important thing to know about the amino acid-derived hormones. 100% of you said C. And the answer that I had was B. So let's go over it. A student is studying an unknown hormone. The hormone is observed attaching to proteins in an aqueous environment. If the student wanted to find the receptor on which this hormone acts, she should look. So A, B, C. A is in the blood. I wouldn't really look for any hormone receptors in the blood. I mean, unless you specifically learned about something that acts on blood tissue, but we haven't really been talking about that in this lecture, so I'm glad nobody picked that one. D, I also would not say. because I just don't think we would find a receptor in the extracellular matrix. I mean, there are, the extracellular matrix has, like, when we talk about juxtaprint signaling, it can act on a cell membrane, but it doesn't really have receptors. So B and C, I guess, were the two top choices. The reason why I would say it's B is because steroid hormones attach to proteins in an aqueous environment. So those are the lipid-derived proteins, and those are the ones that we said attach to albumin in the bloodstream. So the aqueous environment that you find hormones in.
Really hard to stay focused on.
Yeah, so we would most likely observe that this is a lipid-derived hormone, and go through the membrane, and they act on internal receptors. If it were on the cell membrane, it would be a peptide hormone, and since peptide hormones are hydrophilic, they don't actually attach to protein in the aqueous environment. They just dissolve by themselves. So that's why it wouldn't be C, because those peptide hormones are the ones that act on the cell membrane. The steroids are the ones that attach to proteins, and they actually diffuse into the cell to act on those internal receptors.